Namaste students, my name is Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, Women's University, Jaipur. So in this lecture, we are going to discuss about the disease diagnosis techniques. And this belongs to the subject medical biotechnology. So there are many types of diagnostic techniques, and broadly they can be categorized the direct or indirect methods. Okay, so in the direct disease diagnosis, we directly observe for the pathogen. Okay, visually we by using the microscope directly on the slide or after culture, we can observe the microbe or the cause of the disease, specifically the infectious disease. Then some indirect methods are also there, like for example the identification of the proteins, the antigen antibody assays. Okay, so these are the indirect methods. Then one specific category is also there, that is the nucleic acid based identification methods. Okay, so in that we do not observe, we do not look for the microbe or the pathogen, but we find out the specific sequence, that is the characteristics of that particular pathogen. So in that we search a specific gene or the nucleic acid sequence in that. Okay, so we will discuss what are the various diagnostic methods one by one. So the culture is normally the very good identification methods for the organisms, but the culture methods generally they require some time, means the culture requires incubation. Okay, it may be from few days to few weeks, 1 weeks. Okay, so at till that time we cannot wait for the medications. Okay, so the medication should be started immediately when the patient, when the person is suffering from some specific disease. So we need some methods which detect or which can identify the um, cause of the disease instantly or in a few hours. Okay, so for example, the susceptibility assay or the gram staining or the microscopy direct observation. So these are the methods which are very quick. Okay, so first of all we will discuss about the basic technique, that is the microscopy. So what is the microscopy? To use the microscope for the identification of the pathogens. This is the microscopy. Okay, so it is quick, and this is a quick method. Okay, you can observe the pathogens and it will not take much time, but the accuracy of the results depends on the two things. One is the instrument, that is the instrument should be very good, the quality of instrument will be should be fine. And second is the perfection, that is the experienced microbiologist should be or microscopist should be there to give the accurate or the quick results. So the microscopic examinations of tissue may be required to distinguish invasive disease from surface colonization. It is a distinct not easily achieved by the culture methods. Most of the specimens are treated with stains that color the pathogens, because if you do not use the stains, the contrast will not be there. So we use the stains to get a clear picture. The unstained samples can also be used to detect the fungi and certain other pathogens. So the examples of the microscopy are for example, the gram stain is there, that recognize the gram positive and gram negative bacteria. Then the acid fast stain is there, that is generally performed for the Mycobacterium tuberculosis or Mycobacterium leprae bacteria. that are responsible for the TB and the leprosy respectively. Okay, so the procedures for each of the staining is different. For example, if we talk about the gram stain, then we have to first use the crystal violet stain, that is the blue color stain, and that gives the blue color to the gram positive bacteria and not to the gram negative bacteria. Then we use mordant that fix the stain on the bacteria, and then we have to use a second counter stain, that is the safranin. Then the contrast is generated. Gram negative bacteria will be shown as red, and the gram positive bacteria will be shown the blue in color. Likewise, if you talk about the acid fast staining, so it is performed for the Mycobacterium species, and it categorizes the bacteria into the acid fast positive and acid fast non-acid fast and acid fast bacteria. Okay, so it is generally used for the Mycobacterium species. Then moderately acid fast organisms like, for example, the Nocardia species and Rhodococcus and related genera can also be stained. Oocytes of some parasites can also be stained by the acid fast staining. Some of techniques uses the fluorescent stains, and that gives a better image and better contrast of the images. Then many other types of staining methods are also there. For example, the weight mounts is there. Okay, then the India ink stain is there. Then right stain and Jimsa stain is there. Trichrome staining is there. Okay, HNE, hematozylene and hematozylene staining is there. So that are the staining techniques. Various broad categories are there, and we can we have discussed the basic stainings. Okay, so next category is the culture techniques or culture methods. So what are the culture methods? The culture is the growth. You grow the sample which having the microbes, which having the pathogens on a nutrition media. The media may be solid or liquid. Okay, then on the nutrition media, the pathogens grow, and you can recognize them by their appearance, the microscopic appearance and macroscopic appearance. Okay, microscopic appearance, you can use that the microbes from that culture after they grown, and you can see their structure in the microscope. The macroscopic observation is the morphology, size, shape of the colony of the bacteria or the fungus. Okay, so you can recognize the microbes on the basis of the macroscopic observations too. Okay, so this is the culture method, and the nutrition media is vary from bacteria to bacteria, from fungus to fungus. Okay, there are various types of the culture techniques are there. Say for example, the blood cultures. Okay, then the culture methods for the aerobic bacteria, for the anaerobic bacteria, for the mycobacteria. Culture method for viruses is a different technique. Mycobacteria is a different technique. So it have a vast range of the culture techniques. If you want to grow the fungus, then it needs some different types of the media. So each of the microbe need a different types of the media. Okay, so that was about the culture methods. Then the immunogenic tests are also there. Immunogenic techniques are also there to identify or to diagnose the diseases. So for example, you can identify the antigens from the microbe, or you can recognize the antibodies which are generated when someone is infected with some specific bacteria or virus or fungus. Okay, so antibodies are generated against the antigens, and you can recognize, you can identify, you can diagnose the antibodies. Okay, so antigens can be diagnosed, or you can diagnose the antibodies. So one if test is the in the immunologic test is the agglutination reaction. Okay, so in that we use the power of the agglutination, that is the antigen antibody reaction. Okay, likewise the complement fixation is there. So complement fixation measures the complement consuming antibody in the serum or the cerebrospinal fluid. The test is used for diagnosis of some viral or fungal infections, specifically coccidioidomycosis. Okay, then enzyme assays are there. Okay, you have heard about the ELISA, you have heard about the enzyme immunoassay, then precipitation tests are there. Okay, so Western blotting are there. In the Western blotting, you also recognize the presence of specific types of the proteins or the protein stretch. Okay, so that is the Western blot method. So these all are, they have the different specifications, different procedures, and different interpretations. Okay, so one of the new technique is there, that is the nucleic acid based identification methods. Okay, so what are that methods? So nucleic acids are molecules, so it is also known as the molecular methods, and in that you specifically identify the microbe. Okay, say for example E. coli is there, but in it have various strains. You can identify the specific strains using the nucleic acid based methods. Okay, here you use the power of the specific genes that makes them different. Okay, on the basis of our sequence, genomic sequences, we are different, and this uniqueness can be used to diagnose, to know the presence of the specific pathogens. Okay, so say for example the PCR method is there. Okay, RT-PCR that is recently utilized for the corona viruses. Okay, RT-PCR test is there. Likewise, you can recognize the other viruses, other bacteria that is not possible to recognize in the microscopy or other methods, because some of the microbes have considered in a similar family. So they have the similar type of the morphology, they have the similar type of the chemical or the physiological properties. So we have to use the power of the genes or their sequences. So it become complete the in the clinical settings, the results is rapid. Okay, and it allows the patient to be placed on specific antimicrobial therapy and avoid the prolonged management of the empirical potentially inappropriate drugs. So nucleic acid based methods detect the organism specific DNA or RNA sequences extracted from the microorganisms. Okay, so you have to first pick up the sample, then you have to grow it, and you isolate the DNA or RNA, that whatever the genetic material of the pathogen, and then you can detect for the specific genes. Okay, so that was about the nucleic acid based method. So these were the various methods. Many more methods are also there, and that varies, and they are selected on the basis of the requirement. What is the condition, what is the requirement, what is the availability is there, that set our preferences, that which technique we have to use. Okay, so in this we have studied the various methods. For example, the microscopy methods, culture methods, the antigen antibody assays, immunologic assays, nucleic acid based identification methods. So hope you are satisfied with this detailed session. And if you have any doubt, any query, you can write in the comment box. And this session is powered by Digital Version 2, Jyoti Vidyapeeth, Women's University, Jaipur. Thank you.